Okay, my name is Dallin Stokes. I'm one of Dr. Lane's grad students, and today I'm talking about plant viruses because Dr. Lane hates plant viruses, and I find them fascinating, and so she delegated that to me while she's out of town. So I'm excited because plant viruses are bizarre, and so we're going to talk about how they function differently from the viruses you're used to. Okay, let's go over a bit of history first. So of course, plant viruses have been around since time immemorial, but humans have only recently started learning about them and studying them. In the 1600s, they had a bunch of tulips that were infected with what's called tulip breaking virus. Of course, they didn't know it was infected with a virus. They just found these beautiful tulips and decided they were the fad. And in the Netherlands, they had this big craze called tulip mania, where tulips were just the thing. And tulips were going for crazy prices. If you convert then prices to now prices, that's a $250,000 flower for one bulb. So if you think about it with your typical, your average wage earner's income, a $250,000 flower is insane. Unfortunately, with this Semper Augustus tulip, we don't have them anymore. They're extinct because the virus that made them look pretty like that also killed them off. After a few generations, they became less viable until the whole strain went extinct. So not only do you pay $250,000 for a bulb, you pay $250,000 for a bulb that dies. It's a real win right there. In 1892, tobacco mosaic virus, which you're going to be seeing a lot today, became the first virus ever discovered. Now, this isn't just plant viruses. This is any virus. It became the first virus to be discovered, was actually a plant virus. In fact, that's where the name virus comes from, because the gentleman who first discovered them pureed a tobacco plant that was sick and put the liquid onto, or ran the liquid through a filter that was supposed to catch any bacteria, and then put that liquid onto another tobacco plant, and it got sick. So they realized that the liquid didn't have bacteria, but it still caused them to be sick. So we called it poison. Virus, or in Latin, is virus, which means poison or liquid disease-causing agent. So tobacco mosaic virus is actually the source of the name virus. In the 1930s, it was the first virus to actually be crystallized and its structure determined. Before, it was just this amorphous liquid that we know there's something in there that makes these plants sick, but we don't know why. But in the 1930s, they were actually able to see it and determine its structure for the first time. I don't know why they were still doing science during the Great Depression. Seems like they would have better things to do. But we're grateful that they did. Then in 1964, tobacco mosaic virus again was used to verify the universality of the genetic code. What we mean by that is, you know your codon charts from genetics? Everyone's like, oh boy, genetics. Why? Genetics is so much fun. Tobacco, or what they were talking about here is all cells, with some exceptions, use that same genetic table to read their DNA and read their RNA to make proteins. What they proved with tobacco mosaic virus is that viruses do the same. They use the same code when translating their RNA into DNA or into proteins there. So it shows that that same code applies across viruses, bacteria, all kinds of cells. So a bit of background on plant viruses themselves. There are, of course, thousands of different species of plant viruses. I use species, that's what they call them, even though they're not technically in the tree of life. They cause about $60 billion worth of crop losses worldwide. Now, we don't have any plant majors in here, right? I mean, $60 billion is worth a lot, especially if it's taken out to farm field. And it affects all kinds of plants. Here's a couple of different examples of crop species. We've got cassava, which is a major staple crop over in Africa. Like wheat for us is cassava for them. Like cassava is the foundation of their diet. So if it's killing off your plants, like this plant does not look like it's doing so hot, that's a bad thing. Grapes, any wine lovers here or just plain old grape lovers, you can lose a lot of money to, whoops, wrong button, to grapevine red blotch associated virus because it'll kill off your plants and make your grapes taste less good. I read one article saying this particular virus can cost up to $60,000 per hectare if it gets into your fields. So that's a lot of cost to California, particularly because, you know, they both make and drink the wine. And then corn, corn, we all love corn. I mean, anyone doesn't love corn? I don't want to overgeneralize here. That was a corny joke, sorry. Okay, so corn, you can see these corn plants look like they aren't doing so hot. They aren't just dry, it's a virus that's actually killing them off. And of course, our lovely potato, the state religion. This potato looks like you just dropped it on the ground. In reality, it's rotting from the inside out. And that potato picture was taken in Idaho. So this is actually a local problem where viruses can get into your plants and then you're in big trouble. Most plant viruses, it's possible that most plant viruses don't even cause symptoms. So we probably haven't discovered a sizable chunk of them, largely because, for one, most people only care about the crops, right? Like, who cares about the viruses infecting the grasses on the side of the road? We want them to die, right? Like, that's the problem. But with the ones that actually cause diseases in crops, that's harder to, or ones that cause diseases in other species. Anyway, sorry, I'm still waking up this morning. But there are some indications that some plant viruses just hide in their hosts and don't do any external damage or anything like that. So they'd be harder to track down. And some viruses are even shown to be helpful to the plants they infect. For example, they make the plant less desirable to insects that typically try to eat the plant. So they produce chemicals or whatnot that make the plant less edible for the insects that attack them. So the virus can actually be a beneficial infection, not just for all of us medical people where we say virus bad. Okay, so general characteristics of plant viruses. Most of them are rod-shaped. Now, typically what we're talking about in this class, we talk about round ones, icosahedral viruses, but most of the plant viruses are actually rods, and I don't know why. I have yet to be able to find out the reason why that particular shape is preferred. I'm sorry, leaving you hanging on that one. Most of the viruses are single-stranded RNA, particularly plus-stranded RNA, which is your Baltimore class 4s. I don't know if Dr. Lynn teaches you Baltimore classes. I know she hates Baltimore classes. Yes? Okay, good. Baltimore class 4. So like 65% of them are Baltimore class 4. 75% of them are single-stranded RNA. And then double-stranded DNA plant viruses are extremely rare, almost non-existent, actually. And I've got theories on why that's the case that I'll describe later. But again, it's just theories, doctrine of Dallin, right? So we'll share those later on, but leave you wondering here. And what's also interesting about plant viruses is they have extremely broad host ranges. Now, you guys will learn in this class, you know, the host range is usually determined by what? Like, what about the virus determines what hosts can be attacked? Yeah. It's receptors. Excellent. So it can only infect creatures where it's where the spike proteins can match receptors and actually get inside the cell and replicate. Well, plant viruses work differently. They evolve based on the vector, not on the host plant. So a lot of these viruses have extremely broad host ranges, like tobacco mosaic virus. You know, it's got tobacco in its name, so it, of course, attacks tobacco. But it also attacks things from watermelons to orchids, to, like, it's all across the spectrum. It can attack basically anything. And we'll talk about why that's the case. But some plant viruses can only go for 5 or 6 different species. Some of them can go upwards of 100. So it's really the vector, not the host, that is the determining factor. And we'll talk about why that's the case later on here. Most plant viruses, and let's back this up here. I already revealed it. We'll go for it. Okay, most plant viruses lack spike proteins. Now, this is bizarre because we already talked about spike proteins get you into the cell, right? So these guys don't have spike proteins. Now, let's think about this. The whole purpose of a spike protein is to get you inside the cell. What's different about a plant cell versus an animal cell, particularly the outside of a plant cell? What makes a plant cell different externally? Sorry? Cell wall. They have a thick cellulose cell wall. So if I've got an armored cell wall, where are the receptors? There's nothing to attach to, right? If you just bounce off cellulose, binding to cellulose isn't going to get you inside the cell. So these viruses can't get inside the cell in the typical manner that you guys are used to because there are no receptors. So they don't have spike proteins because they're pointless. Now notice I say most plant viruses, and we'll come back to that one. There are some plant viruses that do have spike proteins, but they're not for the plant. So we'll come back for that one. Okay, so we talked about how plant cells have cell walls, so the spike proteins won't work. And of course, transmission is tricky because plants don't usually travel, so they don't kiss fellow plants or hug them or sneeze on them, or right? So you've got to get the virus from plant to plant, and you have to get it inside the cell, both of which are problems. So they have to rely on mechanical vectors to get them into the plant cell. So they have to have something transport them physically, carry them from one plant to another, and then physically punch them into the cell. So there are a couple of ways of doing this. One is, of course, seeds, where the virus infects the plant, gets into the plant's seeds. When those seeds drop and grow into new plants, those plants are infected. But of course, that's relatively rare as a transmission technique, and that doesn't answer the question of how did we get into the original host plant in the first place, right? Like, God said so, it is now in the plant, right? But you, it's got to somehow get in there. In farming, there is vegetative propagation, where, for example, with potatoes, you grow new potatoes by chopping up an old potato, planting each of the little pieces, and it grows new ones. Well, if the virus is in that seed potato, now all of the potatoes that grew from the one you chopped up are now infected. And then grafting is when, like, some fruit trees, like apples, you can take branches off of one tree and put them into another tree. Well, if you take an infected branch off of one tree and put it into another tree, now that tree's infected. So you have to be careful which branches you're moving, right? If it's infected, leave it behind. Nothing? No? Tough crowd. Okay, thank you. I appreciate that. Sad days. Okay. You'll find out I'm really corny. I'm sorry. I don't know why my wife puts up with me. So then there's dissemination by mechanical means, and this one's particularly a problem in farming. Of course, herbivores can damage a plant and then move it along. For example, if I've got a plant like a grass that has a virus in it and the cow comes along and munches on it, the virus gets on the cow's teeth. Now the cow goes and munches on another grass, and that virus now gets dropped off inside the plant from the cow's teeth. So you can have problems like that when you can have contaminated equipment. So in our aforementioned example with the vegetative propagation, if I cut an infected potato and then use that same knife to cut a non-infected potato, the non-infected potato now gets infected because the virus is on the knife. So you have to use sterilized equipment to make sure that you aren't cutting infected potatoes with non-infected ones. Same with shears. If you're pruning an infected tree and then you go and prune a non-infected tree, you can infect the non-infected tree. So you really have to be careful with the equipment. If it can penetrate a cell wall, it's dangerous. You can also have natural plant-to-plant contact, like if the roots grow against each other and rub, you can have that problem. Of course, you have leaves blowing in the wind and they smack into each other. You can get friction there, which can cause kind of surface level damage. And then the biggest one is insect vectors. And we're going to go into more detail there because bugs are by far the most common method of transmission. 70% or more of known plant viruses are transmitted by insects. Of course, major insect vectors include leafhoppers, aphids, and whiteflies. Whiteflies in particular are a problem over in California. But these guys are particular problems. I mean, several of them can fly, and aphids can fly also, sort of, which most people don't know about, but they can blow in the wind. You got a flying aphid, it's Superman. So they can actually, that solves our problem of trees don't move because bugs can move. So if you can infect the bug and the bug goes and bites another plant, now it's infected too. So the general process here is the bug has the virus, it bites the plant, which solves our problem of getting past the armored cell wall, drops the virus off inside the cell, the virus infects the plant, and then another bug comes along, bites that same plant, becomes infected, and drops it off inside the next plant he goes and feeds on, so they can pass it from plant to plant that way. So there are, So that may explain the broad host range. So remember, we were talking about why that certain plant viruses can have upwards of 100 different host species. Now, why would that be the case? Because I remember I mentioned that it evolves to the vector, not to the host. Why would that be the case? What's determining what plants I can attack? Because it's not spike proteins, I don't have those. Yeah, it's the diet of the insect vector. I can get inside whatever plant I can get in, or I can get inside of whatever plant my bug bites. Because most plants replicate DNA and build proteins in about the same way. So the limiting factor is which cells I can get inside of, and the cells that I can get inside of are determined by my bug. So the bug determines the host range. The vector determines the host range, not the virus, which is very interesting. Okay, so types of insect transmission. We're scientists, which means we have to break everything up into categories, right? We have to be more and more and more specific. So there's non-persistent. Non-persistent means that the virus just gets on the mouth of the bug when he bites it, and then when the bug goes and bites another plant, it falls off the mouth and gets on there. Now, it's called non-persistent because you only get so much virus on your face, right? And so then when he can only bite so many plants before there's no more virus on his face to infect. So when a bug bites an infected plant and gets it on his face there, it'll only last for a few minutes before that bug is no longer a problem anymore. So that's why it's non-persistent. We aren't talking about how the virus doesn't last long in the plant. We're talking about how it doesn't last long in the vector, in the bug. Semi-persistent seems very similar to non-persistent, and it took me forever to figure out the difference between these two. It's a bit subtle. Semi-persistent, the bug actually sucks the virus up, and it gets inside the bug's stomach, and then it stays there. It doesn't replicate, it doesn't do anything, it just chills inside the bug's stomach. And then when the bug bites the next plant, it vomits the virus back into the plant. Yeah, isn't that a pretty picture? So, I mean, you vomit on your food before you eat it, right? The bugs say it's a good way to do things. They beat us to it, evolutionarily. So maybe that's the right way to do things. I don't know. I'm not advocating bulimia. We're ISU, I promise. Okay. So persistent viruses, these ones last longer, because with the semi-persistent one, it's going to last longer than the non-persistent one because it's hiding in the gut. And so I'm not going to drop off as many viruses each round because we don't vomit the entire stomach contents into it, right? It's just got saliva and stuff like that that are getting into there. So it'll last longer than a non-persistent one, but it still won't last forever. Persistent ones actually get internalized by the bug. Now, there are two different subclasses of persistent because, of course, we're scientists and we have to categorize everything, and people have to have reasons to publish papers. So we have to come up with new terms. So circulative means that the virus gets into the bug, so the bug eats it, and then it can actually travel across the membrane into the bug's bloodstream, their version of a bloodstream. And then it travels to the salivary glands where it then gets spit back out again the next time the bug bites something. But notice, it's not replicating. It's not actually making new baby viruses. So in this case, it's kind of like the semi-persistent version, but it's going to last a heck of a lot longer because it's actually throughout the bug. It's not just chilling in his stomach. Because I can imagine chilling in something's stomach makes you not last very long. So these guys are unable to actually make new baby viruses. Now the propagative one, these are the guys that have spike proteins because they do have to get inside the cell, and bugs aren't plants. Who knew, right? The one thing you learned today, bugs aren't plants, right? Put that on the exam. So the propagative ones are actually able to replicate both inside a plant and inside the bug. So they have spike proteins, but they're for the bug's cells. So it's actually both an insect virus and a plant virus at the same time. It's able to replicate inside both. Now these ones last a really, really long time. In fact, they can actually last upwards of the insect's entire lifespan because they usually don't harm the bug. The bug is the mailman. He's not the target, right? Oh, dang, now we've got terrorism. I'm sorry. ISU's going to throw me out. We're going to, right? So so they usually don't infect they usually don't sicken the insect. They just replicate inside it, and then they get into the plant where the bug bites, drops it off, and that's where the problem actually happens. Because if you kill your bug, you don't get anywhere. So bugs we like, plants we don't, right? Again, I'm expecting people to rappel through the window any minute now. Okay, so virus replication, we're going to kind of blow over the molecular details of this because they're very similar to what you're already familiar with, right? They're plus-stranded, single-stranded RNA viruses. They're going to replicate the same way that you're familiar with. Some of them have hairpin loops, some of them have poly A tails, some of them have, right? They're going to behave like you're used to. There's nothing new there. So rather than repeat everything you've already learned, we'll go into the stuff that's kind of unique here. The method of uncoding is still unknown. Now, this may be surprising, but if you think about it, how does a virus usually uncoat? What makes a virus capsid break open typically? Have you guys learned that in this class yet? I don't know how far in the semester you are. So. Oh, okay. So typically, right, they get endocytosed, which puts them inside an endophagosome. It merges with a lysosome. Typically, like a pH drop will come in and crack the thing open, right? Well, there's our problem here. We're not getting endocytosed. The bug bit the cell and dropped me off directly in the cytoplasm. So there is no pH change. There is no lysosome. There's nothing like that to uncoat the virus. So we have yet to figure out how it uncoats, but somehow magically it does. So that is still an area of research. Part of the problem is, plant viruses aren't exactly the big money area. Persuading a multimillionaire to invest in plants is, they usually get left behind. So the method of encoding is still unknown. Usually what you're used to here, the reverse transcriptase comes in, binds to the endoplasmic reticulum, the RNA dependent RNA polymerase comes and binds to cellular membranes within the cell. It typically binds to the endoplasmic reticulum, but it can bind to any membrane within the cell. And then it folds the membrane out into a bubble called a viral replication complex. Have you guys talked about those yet? A little bit? Okay. So most of the ones that you've talked about, they form a viral replication complex by building it out of proteins themselves, using viral proteins. This one, typically in plant ones, they actually build it out of host membranes. So they'll invaginate it out, build themselves a little bubble of membrane, and then put their polymerases and all their replication complexes within the bubble. Now, why might that be advantageous? Sure. Yeah, go for it. Absolutely. Good. Why might it be a good idea to have my replication complexes in a bubble? Why not just replicate in the cytoplasm? Everything's not spreading out. Excellent. So it's essentially a factory, right? Like it's the benefits of having a factory versus home industry, right? You can concentrate resources, you can concentrate DNA or RNA nucleotides, you can concentrate all your enzymes in one place. So you can manufacture a lot more viruses a lot quicker in this case. So this picture here is actually an electron micrograph here. So you'll notice in purple, this is a mitochondria. Trust me, it doesn't look like one, but it is. And then in yellow, you've got the virions. In this case, they are round viruses. And then over here in white, you've got this viral replication complex bubble, and it's full of little baby viruses. Now, it's not obvious here, but this bubble isn't complete. There's actually a hole in the side of it that allows new resources to come in and baby viruses to go out. Now, what's interesting about this is how do we get, oops, it was delayed, there we go. How do we get from cell to cell? Because, you know, in an animal or in a human, you spread from cell to cell by bursting out of the cell and then binding to the cellular receptor of the next cell along the line and get inside. But again, we don't have spike proteins and we don't have receptors. So the bug drops me off, but he's only going to infect 5 or 6 cells. So how do I get throughout the rest of the plant? The trick here is plant cells have what are called plasmodesmata. There's a tongue twister for you. They're these tubes that link each of the cells to another. So essentially, a plant is one big cell, if you think about it, because all of the cells are interlinked. So if you can get in one cell, you can get into them all. So they don't need spike proteins. However, if you were to just diffuse across or through the plasmodesmata, it would take you all year to infect that whole plant. And you want to infect the whole plant because you want to increase the odds you're going to find another bug, right? If I'm only in one leaf, what are the odds that another bug is going to come and bite my leaf, right? I've got to kind of spread out so that if any bug bites my plant, then I have a chance of getting picked up and shipped off to another plant. So we have to move from cell to cell as fast as humanly possible, or virusly possible. So the way they do this is they utilize the cell's motor proteins. So if you guys have taken cell and you know about dynein and kinesin, which are proteins that can walk along microtubules. For all you medical people, it's myosin, right? They're the motor proteins. So the viruses build these proteins that are capable of sticking the entire virus replication complex. We're not taking one virus, we're taking the factory. We attach it to one of these transport proteins, and then the transport protein walks me to the plasmodesmata. Now, the problem there is plasmodesmata are too small for my viral replication complex to fit through. That's a problem. So the viruses actually produce a protein called a viral movement protein that actually goes out and physically makes the plasmodesmata bigger. all of them. It's very efficient. They've done tests on these where it basically finds every single plasmodesmata in that plant cell and makes them all bigger. And they can see this travel outward in a ring. So they used fluorescent proteins on these things to watch it. And it actually travels outward in a ring, making every plasmodesmata bigger. So once it's bigger, the protein can just walk us right through. And ta-da, I've brought the entire factory into the next cell. Now, what's nifty about that is, unlike with animal viruses, where you have to build an entire virus to be able to infect another cell, in this one, your viruses, you just need the RNA, right? You don't have to complete the process before you can infect the next cell because the entire factory is going. So this process is extremely efficient. You only need 1 to 15 copies of mRNA to jump into the next cell to make that cell infected. It's very, very efficient. And of course, we're taking the whole factory with us, so we're going to achieve that limit very easily. I don't know. I thought that was kind of fun. Here's a little experiment. Now, this photo looks sketchy, and it is, because it's a photograph out of a book from like the 1930s that I found in the Oboler Library, and it had about this much dust on top of it. So I can tell that people really read about plant viruses at the Oboler Library down there. So this was an experiment that was done in the 1930s, and this is kind of a diagram of a tomato plant. And what's going on here is they infected one leaf. So it's this little striped one here on the first panel. The scientists took this tomato plant and they infected one leaf on that tomato plant. And of course, they did this to thousands of tomato plants, I'm sure, because we need to have lots of replicates. And then after each day, they would take one tomato plant and basically shred it. So you slice it up into pieces, because they didn't have electron micrographs or anything like that, right? So they didn't have any way to actually visualize the virus moving through this thing. There aren't fluorescent proteins. There aren't anything like that. So these guys were smart and figured out a way to do it. You slice it up into pieces, you puree each piece, and then put each piece on and culture each piece on a petri dish and see which ones come up sick. If it comes up, then that piece was infected. If it doesn't, then the virus hasn't gotten that far yet. So for 3 days, the virus just stayed in that leaf. Because remember, we have to travel through plasmodesmata, and that's a little slow. So we're spreading out throughout the leaf over the course of 3 days. Now on day 4, this really takes off. We suddenly go from the leaf to the roots in 1 day. Now, it took us 3 days to spread throughout one leaf. How the heck did we get to the roots in 1 day? Anyone remember their plant stuff from 1101? Sorry? Do tell. So plants have essentially blood vessels called xylem and phloem. So the phloem is responsible for carrying nutrients down to the roots, and the xylem carries it to the top, if I remember correctly. I didn't get those backwards. I'm sorry if I did. One of them carries stuff down, the other one carries it up. So you travel, it takes you 3 days to travel throughout the leaf. But then once the viruses get into the phloem, now you've got a highway. You can travel down the highway and drop down into the roots, and ta-da, you can infect the roots down there. So as soon as you can actually hit the phloem, you're home free. And then within a day, it travels from the roots, hitches a ride on the xylem, and shoots to the top. Now notice, we skipped everything in between. So the black leaves are the infected ones. So we went specifically to the young leaves. They found that old leaves are actually resistant. The virus prefers young leaves over old ones. And I don't know the reason for that, and I'm sure in the 30s they didn't have the technology to find out the reason for that. I guess maybe young leaves would be replicating more, but later tests found out that the plant's version of stem cells, which would be replicating very quickly, are also resistant. So it's it's not the super fast guys and it's not the old guys. We prefer young leaves. And those are typically found at the top because, plants grow up. Then after 10 days, it's starting to infect the older leaves down here. And then, of course, it's spreading more and more throughout. And finally, after several days, we infect the entire plant. Now, what's interesting is on younger plants, it never gets to the older leaves. So this is on like an old plant. If it gets infected, then eventually we'll get to the old leaves. If it's a young plant, we never bother with the old leaves. We only go for the young ones. Maybe insects prefer young leaves. It's nice and nutritious, right? Like I prefer young leaves on my lettuce. But I honestly don't know why that's the case. But over a period of several days, the virus is able to travel throughout the entire plant. And it's not just Plasmodesmata to Plasmodesmata. Questions so far? All right. Sorry, I've been kind of blowing through this. Okay, so plant viruses, we've talked about how they cause diseases. Of course, we have to try and find ways to fix that because, $60 million, people, yes, billionaires don't usually invest in plants, but somebody does because there's $60 million worth of crop losses, and that's a lot of money. So there are efforts to try and find ways to fight back. Now, the problem is once a plant gets infected with a virus, that's game over. There is no treatment for a plant virus. So the main way is prevention. So, you can spray for insects, which kind of works. It doesn't perfectly work, but it kind of helps. The main way that's been developed is producing varieties of plants that are resistant to the viruses. This, you can do it through typical breeding. You can find a variety that naturally develops virus resistant, breed it with your crop version to create a version that has the trait of being resistant to said virus. So now, even if the bug bites my plant, the virus will get in there, but the plant is actually able to defend itself and kill it off. But the up and coming way is to do this through genetic engineering because now you don't have to do crossbreeding over several generations. I can just take the trait I want, put it into the plant, plant's invincible, right? So here are a bunch of different plant species that they have toyed with here. Papaya, which I'm going to use that one as a specific example here in a second. Plum, they made it resistant to plum pox virus, which I kind of want to get plum pox because grow plums or something. I'm sure it wouldn't be pretty, but it would be fun just to say plum pox, right? And then you've got beans, potatoes. The potato virus Y, now these viruses are put into families, and potato virus Y was given the unfortunate family name of potty viruses, P-O-T-Y. So, and of course that one's a problem in potatoes, and so, it's potty humor there, I guess. But, and then maybe that joke will go viral. And then of course summer squash, sweet pepper, and tomatoes, which we talked about earlier. Now notice the tomato one, here's another example, is named after cucumbers. So even though it's named after the cucumbers, it can also infect tomatoes and be a problem over there. So they can make versions resistant there. Now, papaya ring spot virus, I'm going to use this as a specific example so that we can dive into the actual details. How am I doing on time? Okay, we're good. So we can dive into the actual details of one specific disease just to give you a typical example of how this works and the problems it can cause and how we fix them. So papaya production is a $15 billion industry worldwide. Who knew? A lot of papayas are produced in China, Mexico, Brazil. In the United States, most of them are produced in Hawaii. And the story that we're going to tell here is based in Hawaii as well. In the 1980s, this virus called papaya ring spot virus, or PRSV, ring spot, yes, PRSV. Um, I mean, it's always been in the papaya plants, but it wasn't really a problem, right? Like if the virus infected your plant, you chop down the tree, you burn it, you're good to go, right? Well, in the 1980s, it mutated and became this super virulent strain that just really took off and started killing trees like crazy. And so the only way to try and treat it is chop down the tree and pray the aphids didn't bite your tree and spread it to your other trees before it actually got there, because it causes a lot of problems. And I think I'll get to the symptoms later on here, but it causes a lot of problems to the papayas. And of course, you can only chop down so many trees before your papaya orchard is no longer viable. Well, all the local aphids are infected. So you have to pack up your farm and move to a new area that doesn't have papaya ring spot virus. And of course, there's only so much land, especially in Hawaii, right? You can drive around the thing in 3 hours. So this was a serious problem. And by the mid-1990s, it had basically wiped out the papaya industry in Hawaii. There was one valley, I think it was on Oahu. There was one valley that didn't have the virus. And so that's where the entire papaya industry in Hawaii was. And then the virus got into the valley. So everyone was like, flip, we've got to figure out a problem or a solution to this. And so the University of Hawaii developed a GMO version of the papaya that's resistant to the papaya ring spot virus. Now this one's kind of a fun story because usually it's big major companies like Monsanto and people like that that are coming up with these GMO plants. But in this case, it was a university that did it, and they gave the seeds away for free. They did not patent it. They handed out the seeds and saved the industry. So now they've got these resistant versions. Now what's interesting is the resistant versions are only resistant to the Hawaiian version of the virus, not the Guam version of the virus. So if the Guam version ever gets to Hawaii, they're going to be in trouble again. But for now, it's been great for the last 3 decades, right? 2 decades? Oh, I'm getting old. That's not good. I was born in 96. This is not good. Basically it's really bad. Afterwards, if you guys want to go through, you can watch this video that just kind of introduces the problem. It's from they interview an actual papaya farmer that went through it. So the virus is called papaya ring spot virus. It's from the Potyviridae family. So it's related to potato virus Y. It's a plus-stranded ssRNA virus, Baltimore class 4. It's naked, so it doesn't, most plant viruses don't have membranes. They are just solid ones. Probably makes them more resistant to the shearing processes when getting bit by a bug and dropped off by a bug. And they're typically flexible. See here. So papaya plants are the primary reservoir, but it can infect anything in Cubertaceae. I probably pronounced that wrong. So watermelons and cucumbers, who knew they're related? That's kind of a fun trick. Vegetables are related to fruits. Who knew? Pumpkins and gourds can also be infected as well, but the big reservoir is papaya. And aphids are the primary insect vector here. And of course, the problem there is they can blow from tree to tree to tree. So they can infect an entire orchard pretty quickly. And of course, they can also be spread by pruning the healthy plants. Because remember, one way to treat it is chop down the tree and burn it down and pray that you... So if you chop down the tree, that thing that you chopped it down with is now contaminated with the virus. So now you can't use that to trim other trees or anything like that. So the symptoms, this is what it does to the plant. So clearly that plant's not very happy. And the leaves, they kind of curl up upon themselves and wither a little bit. Now, plant fruits become sweet by the sun running into the leaves, which produce sugar, and then the sugar gets shipped to the fruits. If you don't have leaves, your fruit doesn't become sweet, it stays bitter. So the papayas from this don't taste very good, which means they're hard to sell because people like good tasting papayas, not bad tasting papayas. So the major problem is the damage it does to the plant itself, but it also harms the fruit. And of course, it puts these little rings all over it, which is where it gets its name for ring spot virus. And it makes the plants grow shorter. So it damages the plant over time, which can make it tricky to find out which of your plants are infected, because it's not like the plant just keels over overnight. And so the aphids have time to bite a plant before you even know it's infected, which means you can have a whole orchard of infected trees and not know it until it's too late. There's a big problem. So the treatment is basically wipe out the forest and move. So we talked about how the GMO varieties there. 76% of Hawaiian papaya is now the rainbow GMO variety. I was looking, Walmart doesn't sell it, but it's like if you get papaya in Hawaii, it is basically rainbow or sunup, which is the other brand of genetically modified papaya there. So it's basically taken over the industry down there because it is resistant to this virus, which is still a danger down there because, I mean, it's still down there. So, and